Good evening, everyone. I'm Anthony McAdoo, Executive Director of the WKU Alumni Association, and it's my pleasure to welcome you to our first WKU Expert Exchange with a multidisciplinary look at the COVID-19 pandemic. Tonight, I'm joined by 5 of our outstanding WKU faculty members whose field of expertise offer a different perspective on the current COVID-19 pandemic. These faculty members are here tonight to share their unique insights with you, our alumni and friends, much like they've been doing with our students in the courses they teach and the research they're conducting. I'll now invite each one of our faculty members to briefly introduce themselves and tell you a little bit about their field of expertise. After we complete our introductions, we'll have 3 rounds of questions that have been informed by our alumni and friends who submitted questions as part of pre-registration. During the program, feel free to submit any additional questions or comments using the features of Facebook or YouTube, wherever you're viewing tonight's broadcast. After our 3rd round of discussions, if time permits, we'll entertain some of those questions and a question and answer session with our faculty members. So now, let's meet our 5 faculty experts, shall we?
Hi, I'm Bruce Kessler. I'm a professor of mathematics at WKU, and I tend to work most recently in modeling real-world situations.
Hi, I'm Kevin Williams. I'm a professor and department chair in the Department of Chemistry, and my research interests are in the area of drug design.
Hi, everyone. Rodney King here in the biology department. My research interests are actually bacterial viruses and studying how host-parasite interactions and so forth.
Hi, I'm Susan Eagle. I teach in the public health department here at WKU, and I have a background in anthropology, and my interests are the social and cultural impacts on health.
Hi, I'm Audrey Jennings. I am Associate Professor in History and also Director of the Office of Scholar Development, and my work focuses on history of disability, history of disease, and policy in the United States.
Well, thank you all for joining us this evening. So we'll start our first round of conversation and we'll ask Dr. Kessler to get us started.
Okay, so I want to talk about 2 things very quickly, and that is exponential growth and flattening curve, because these are 2 ideas that don't necessarily go together. And so let me say just a little bit about them and how they fit together in this situation. Most people think of exponential growth as really quick growth. And that's true, it really is. Although it's the starting growth of an exponential curve may look very similar for curves that have different rates. It starts out very slow. And so what I've shown you here are 2 exponential curves. There's a red one and a blue one. The blue one looks almost linear. The red one looks a little curvier than the blue one and starting to grow. The point is that looking at data, trying to decide which of these curves you're on, it's very difficult, because they're so close to each other. In fact, I think in our Excel-minded world, many people would be tempted to do linear regression on these and find the line that matches them. So after 50 units of time, the blue one's at 2.25 and the other one's at 31. Still fairly close if you're looking on a large scale. What happens with exponential growth is that once it decides to start growing, it grows very quickly. And so even this blue curve, if I left it long enough, would grow very quickly. But I show this to you to show that very small changes here can make very large differences here. And so it's very easy in this kind of situation to think, oh, we're on one curve, the blue curve, and actually be on the red curve, which could lead to this happening. The other thing is you may be on the blue curve and we relax conditions and raise that growth rate and end up on the other curve. The growth rate for the red curve, by the way, is about 7%. The blue one's about 1.7%. Okay, so what does this have to do with flattening the curve? The thing about exponential growth, that goes on forever in this direction, and we keep seeing these ideas of getting that curve to flatten and come back down. So to really understand this, I need to introduce to you more than just the infected, which is what I was showing you, but what's called the 3-compartment SIR model. So the exponential growth that you see, we're looking at the rate of change of that infected number, and we're saying that that's related to the number of people infected. The more people you have infected, the faster it's growing, right? And this beta that I put in there is the transmission rate. Well, we don't have an infinite number of people. So we kind of calm that down by talking about two other groups. We have a group of people who are susceptible to catching the disease, and we have a group of people who are removed, and we put everybody in one of those three categories so that the sum of the three numbers is the total population in. And then so we take that rate and we kind of condition it by the number susceptible over the total number. When the total of susceptible is pretty much everybody, then you're just multiplying by 1 and you have basic exponential growth. And you'll see this in the curve. It starts out looking exponential. But, and so the susceptible is decreased, the rate of susceptible people is decreased by that same amount. The other thing that's happening is infected people are being removed by doing one of maybe three things, I guess, maybe more. They are getting better, and so we hope that they have immunity to it at that stage. I say we hope. We're going to talk about that later in the broadcast. Or sadly, they may pass away. People do die from COVID-19, and that's another way to move into the removed category. The other one is they could quarantine themselves and take themselves out of the population so they're not infecting other people. So those folks are leaving at a certain rate, and I've called that rate gamma. Okay, so just like before, it's kind of a scaled version or a constant version times how many are infected, and that rate of removal then. So if you notice, these things all add up to 0. It's kind of a factor of these things adding up to a constant. Okay, so two things here, the beta and the gamma. The gamma is really out of our realm of control. It's kind of has to do with the disease and the treatment of the disease. The transmission rate is the thing we can control. So this is more math than most people want to see. What I really want to show you, let me get out of this, is this model. So the black curve here is the number of infected folks. The red curve is the number susceptible, and the green curve is the number of removed people in this thing. So what we control with beta, how we control beta is things like wearing a mask, social distancing, not going to events with lots of people there, washing our hands. So when we bring this beta down, there's two things going to happen. I want you to watch carefully. As we bring the beta down, I want you to see the effect on the infected curve and also take a look at the susceptible curve.
Hey, Bruce, real quick, not to interrupt you. Don't forget to hit screen share on your, and I'll add that to the stream.
Oh, did I not share anything? I was talking the whole time.
No, you're fine. You're very descriptive in how you talk. So.
Oh, great. So, yes, I'm sorry. I should have hit this.
No
I should have shared my screen, and I should have shared the whole screen. Okay. So I had some cool stuff here. I'll show you later. But I want to show you this. So let me go back. Here are the three curves I was describing to you, right? So the black curve is the infected folks. As we bring the beta down, you can see that the curve is getting flatter. And let me stretch this out for a little bit so we can see more of the effect. So not just is the curve getting flatter, but also the number of susceptible people is it's leveling off. And so what happens in a situation where we control that beta is, okay, so I'll leave it at that. I've kind of overshot my time. Sorry about that. And I'll turn the screen back over to you guys.
All right, hi. So I'm going to talk a little bit about why you might be seeing some changes in our adjustments in what you're hearing from the scientific community or from public health officials right now. And I'm going to start off and just give you a little bit of an overview of how the scientific process works. This is basically, by design, the scientific process is something that's iterative or cyclical. And so you start off making some observations. Based on those observations, you come up with a hypothesis, an idea, basically a tentative explanation. And that hypothesis is going to be tested by experiments. And depending on the results of those experiments, you're going to make new observations that are going to lead you to either revise that hypothesis or advance it further. And if you advance it further, you do additional experiments. And as you go through and do these different cycles, you're eventually going to get more information. You're going to get more confident about your analysis. You're going to get a better idea of an answer to a key question. And when you feel like you've got enough of a question, or enough of an answer to a question, you're ready to try to get that out to the public. And the best way to do that is through some sort of publication. Typically, if we submit a publication, we're going to, first of all, submit it to a journal that's going to send it out to probably 3 or 4 other experts in the field. They're going to look at your methods, they're going to look at your data, they're going to look at your conclusions, and just see if it makes sense. Is the data legitimate? Are the experiments well carried out? Do your conclusions match up? And if they agree that it is, they're going to recommend publication. If not, they're going to recommend modification or even rejection. So let's say the paper gets published. Well, now it's going to go out to the general scientific community, and all the scientists are going to have access to your information and your data. They're going to follow that up. They are going to be able to either advance it further, they're going to be able to challenge it in some way, come up with an alternate explanation. And so what sometimes happens is something will get published, and then later on people will find out, no, that's not quite right, or they overlooked this alternate explanation or whatever. Well, normally that's going to happen kind of behind the scenes, and after a couple of years when the science settles, maybe the public will hear about that. But in this particular case, what we have is a situation that is so urgent, it's leading to mortality, it's leading to morbidity, it's leading to economic impacts, it's leading to stress and mental anguish, that everybody's invested in this. So you've got lots of people doing lots of experiments all at the same time, and people are kind of rushing to get their information out there. And as soon as they do, often the public's finding out about it, and public health officials are making recommendations based off the latest data. But sometimes additional follow-up studies might sort of alter the direction of that recommendation. And so we've seen that happen a couple of times. Probably one of the most well-known of those was hydroxychloroquine. There was a study from France that came out and suggested that this drug that's already available for treating other conditions might be useful also with COVID. And so they put out an emergency authorization and started recommending that hospitals try this in certain cases. And as this additional data was collected, people decided that it wasn't really doing a whole lot of good, and people started to question some of the methodology of the original study. And so the public health recommendations have backed off from hydroxychloroquine. This is not a conspiracy. It's not something that indicates people didn't know what was going on. It's just this is how science works. We make moves, we make adjustments, and then we respond to the latest and best data that we have. If you look at what's happened over the last few months, we've really seen a lot of advancements. We know a lot more about this disease than we did 10 months ago. And even now, if you start looking at people who are hospitalized, the number of people in the hospital who die from COVID is dropped by a factor of 3 or 4. Some of that may be because the population's a little younger, but at least some of that's because we know better how to treat this disease going forward. So as you start seeing adjustments made to the science, that's a natural part of the process, and that's indicating that science is working exactly the way it's supposed to.
All right, hi folks. I'm going to share my screen, pull up a few slides for you. Okay, I hope you can see that. Someone let me know if you can't, okay?
You're good to go, Dr. King.
Excellent. All right, so it's great to be here tonight, folks, and I've been asked to tell you a little bit and talk a little bit about, give some background on what coronaviruses actually are. A little plug for viruses. They're integral to life on this planet. Every living organism on this planet is actually susceptible to viral infection. They have clearly impacted the evolution of life on this planet, and understanding viruses helps us understand pathogenesis. It helps us understand our own immune system, and it helps us understand how to use them for things such as therapeutics. So basic research into understanding viruses is a very important part, a very important endeavor. Okay, so coronaviruses, as you have heard, the name corona comes from the particular type of appearance they have on the electron microscope. And so I've just given a diagrammatic little schematic up here at the top. These little spikes on the outside of the virus give it the appearance of a corona, crown-like looking appearance. And so a couple of other aspects of this virus that you need to understand. When we talk about viruses, we organizing them into groups according to their genome and what kind of genome they have. So there are two basic information carrying nucleic acids. That's DNA, as our cells, as our genetic information is encoded in DNA. There are viruses that have DNA genomes, and there are viruses that have RNA genomes, such as coronavirus. And you may have heard the term positive sense. And what that means is this virus's genome can immediately be translated into proteins. And that's kind of illustrated on the figure here, where the genome is shown at the top, the entire genome. Below that are two large proteins that are made almost immediately after this virus infects the cell. And then these large proteins are then processed into the smaller proteins you can see numbered there. And so there's a very important protein that's responsible for that, and that's a protease, which actually turns out to be a potential target for therapeutics. So these viruses, as most viruses, they're going to interact with a receptor on their host cell. And that receptor has been identified in this case, the angiotensin converting enzyme, which is a protein that sits on the surface of lots of different tissues in our body, okay, including our respiratory tract. And so this virus, surrounding this virus, it has a membrane. And that membrane came from the previous cell in which it infected. And in that membrane have been inserted these spikes. And I know you've heard that term before. So these spikes are what actually interact with the receptor on the surface of the host cell. This entire particle is then taken up by the host cell. And then there's a membrane fusion event that allows the viral nucleic acid to be released out into the cytoplasm. And this is where it has access to the cellular machinery of the cell. And all viruses require that. All viruses are parasites. They're obligate parasites, meaning they have to have a host cell in order to make more of themselves. And so lots of events happen, as you see numbered in this diagram. And all these events are potential targets for therapeutics, in fact. And so different viruses, more or less, is known about these different steps. But of course, the viral nucleic acid has to be copied to make more copies. And eventually those are packaged into viral particles and they're released. And those particles can go on to infect additional cells or more cells. So again, these different steps, we've learned so much about these steps over the last 6 or 8 months. And a lot of that has been facilitated by our previous knowledge of other viruses and similar steps that those do. So those are the basic steps of what the virus does. Again, this envelope is derived from the host as it exits, okay? And so envelope viruses tend to be a little more fragile, if you will, than viruses that do not have an envelope. And this is one of the reasons why things such as our alcohol gels that we use to sanitize are effective against viruses of this type. So, as I mentioned, the receptor is on the surface of these cells, but recently there was a paper, very recently, that they discovered another receptor. And this is thought to maybe contribute to increased infectivity. So this is an important discovery, and there are other viruses that have a similar type of setup in terms they have a receptor and they have a co-receptor. So this particular receptor, neuropilin, is actually absent from the original SARS-CoV, that is the SARS virus that was discovered back in 2002, or the outbreak in 2002, 2003. And so this protein or this virus has this ability to infect or actually attach to this other receptor on cells. So this is thought to increase its infectivity because you have another binding interaction for that. Including what I said here on this slide, neuronal cells facing the nasal cavity are positive for this particular receptor. I also just want to briefly mention the fact that those steps that I talked about in the previous slide, one of the aspects of this particular virus, it is an RNA virus, all right, but coronaviruses are the largest of the RNA viruses, and genome size, in this case, is thought that these extra genes, if you will, contributes probably to the ability of the cell to deal with the immune system and other ways of surviving and replicating in host cells. So this particular virus is quite stable, interestingly, but I did want to show this evolutionary relationship here. SARS-CoV is here, which turns out to be a novel virus, okay, because it is more dissimilar from the original SARS virus discovered several years ago. In fact, one of the closest relatives of SARS-CoV, COVID-19, was actually isolated from a pangolin, an animal that in China that was isolated from that particular animal and found to have very close, over 99% or so similarity. Another type of coronavirus that was responsible for outbreak in 2012 is MERS, okay? And that outbreak occurred in the Middle East, and from this analysis, you can see that that's even more dissimilar from SARS-CoV and even the original SARS. So they're all part of coronaviruses, but there are enough sequence differences that that SARS-CoV or COVID-19 is a separate, deserves to be in a separate group by itself. The coronaviruses showed down here at the bottom are coronaviruses that have been known for quite some time, been circulating in the population, and they cause mild symptoms, okay? These particular coronaviruses don't cause just mild respiratory symptoms. Just quickly, the closest relative of SARS-CoV or COVID-19 would be a virus isolated from a bat, 96% identity to that particular. So this is thought to be how the virus is maintained in the environment or in animals. And so it's thought that this virus originated in an animal, meaning that's a zoonotic disease that originated from an animal and was transferred to humans. I'll skip that one, and I'll skip this one, and I'm going to end there and allow the next person to go.
Hi, so I'm going to address a question that was sent in ahead of time, and the question is, what are some changes that WKU can make to improve safety of students, faculty, and staff? So more of a kind of a prevention, public health kind of question. First thing I want to say is that we're doing pretty good, right? We've done a lot of the things that we know need to be done in sort of congregate settings. So we limited class sizes. We've pivoted online when classes were too large. We are using other modalities, hybrid, etc. And so we're good at limiting the number of folks that are congregated in enclosed spaces. We're also pretty good about mask wearing. We're good at limiting gatherings, though we can see as you walk around campus, you can see some fatigue, mask fatigue, and sort of general tiredness with the situation creeping in. But on the whole, we've done well, and our numbers are fairly low compared to the county and to state levels. Now, what could be improved? There are a couple of things that we can improve, and these are improvements that are focused on best practices during pandemics or healthcare emergencies. Communication is key during health crises or pandemics like this. Really what we want to do is make sure that we're pushing information out to everybody rather than just having information there to be discovered. So we really want to make sure that we're very open, transparent, that we have regular updates for information. And Governor Bashir is a really excellent role model for good, clear, regular, transparent information sharing. And also doing so with in a very human way. And you know, we can all sort of recite his catchphrases. You know, we're in this. We're going to get through this together. And so he provides an excellent role model for communicating serious information and doing so in a very human way. Another way that we can sort of foreground communication and make sure that we're getting information out to folks so they can make the best informed decisions possible is perhaps modifying some of the case dashboard features. Our case dashboard does give us the basics. It could be updated more regularly, perhaps daily. It could include past information so that trends over time are visible, so that we can see from week to week, you know, have cases risen? Are they falling? just so that we can see trends over time and we can respond accordingly. Perhaps indicate things like how long the wait is for tests, for results, how many students are in quarantine or in isolation, how many faculty, how many staff members are in quarantine or isolation. It could also include county-wide information, since our university is so strongly interconnected with the community, making it very easy for students, faculty, and staff to know what's going on in the broader community is going to give us the information that we need to help us assess risk as best we can. And for some good ideas of what is possible and what can be recorded and reported on dashboards, there's a website that sort of aggregates information about different university dashboards rather cleverly or transparently actually named ratecoviddashboards.com. And it gives you a good idea of what different universities or choose to include to make sure that their campus community is as informed as possible. And then, of course, the gold standard for universities for helping to increase safety is to shift from the operative mode of just testing those who are symptomatic or those who are known in the community to have had contact with positive cases, to shift from that to surveillance style testing, which would be regular testing of different samples of the population or the community, rather than just reserving tests for those who are symptomatic or have known contact. This allows us to catch pre-symptomatic cases and allows the community to get in front of any potential outbreak as opposed to catching up after the fact. Now, the downside of updating the case dashboard on a daily basis, doing surveillance testing as opposed to symptomatic testing, is that it's expensive. It takes a lot of time, a lot of resources. And like I said, on the whole, we're doing well. Is there room for improvement? Well, there's always room for improvement. But as long as we keep doing what we're doing, as long as we keep from letting that fatigue get to us, and as long as we are, doing what we're supposed to do, wearing our masks, keeping our social distance, we should see low rates continue. Thanks.
Hi, so I got a question that asks, is there anything we can learn from history about the long-term social and public health consequences of pandemics that continue to affect societies even after the pandemic ends? And what sorts of measures can be taken now to mitigate those consequences? And so I love this question because I think that rightly so, a lot of the discourse right now is focused on what's happening right now. So where are the numbers right now of infections, the deaths right now, the drive for a vaccine, the search for effective treatments, what's happening with the economy right now. But I think that we're at the beginning of a much longer story. And so I think that the past can give us some context for where to look and what to kind of predict. And so we don't fully yet understand the long-term consequences of COVID-19 infection. And so I think that's an important piece. So the CDC suggests that there's a relatively long list of somewhat common complications, many of which have the potential to create long-term or even lifelong disabilities. And so I think that raises questions about our healthcare capacity to provide needed and perhaps long-term services. So we focused a lot on, the immediate concern about the surge, but what's that long-term ability? And then also, what's the long-term economic impact? And so the Spanish flu pandemic of 1918, 1919, I think can give us some ideas about where to look. And so that particular pandemic was particularly disruptive in working class homes, where prolonged illness or death might spell economic devastation or even the collapse of a family as children were moved out of the home because of death or prolonged illness. And so most of those deaths during the Spanish influenza pandemic were among folks who were aged 20 to 40. And so it was unlike many other flu epidemics in that it was more likely to cause death among that sort of younger population. And even with that said, we saw a lot of excess death in very young people. And that death wasn't necessarily coming from influenza, it was coming from other illnesses. And so there was a spike in susceptibility to other kinds of childhood illnesses, particularly those that were respiratory illnesses. There were a lot of deaths related to kidney disease, to diabetes. And so essentially many people were dying kind of around the pandemic, indirectly connected to it. And we're already starting to see that with the current pandemic. But I think that we'll likely see increased disability rates, and those disabilities may have direct or indirect connection to the current pandemic. And so I'm going to talk in response to a question a little bit later on about the economic consequences of the Spanish flu pandemic. And so I thought right now I would talk just a little bit about the economic consequences of reoccurring polio epidemics and really widespread disability. So in 1930s United States, working conditions contributed to disability, various childhood illnesses. Without antibiotics, even what today we would consider relatively minor infections could lead to disability. Also limited access to health. And so in a lot of ways, we see a whole lot of New Deal policy responding to that excess disability. So some of that policy we don't necessarily think about as a response to disability, but I very much think that there's a really strong connection. So if we take something like the Social Security Act, so when we think about Social Security, we think about old age pensions, we think about unemployment insurance, and those were certainly important pieces, but it also included funding to provide health care for disabled children. It included funding for public health work, for rehabilitation, for aid to blind people. And really in the original act and the people who were developing it, there's a strong sense of disability connected to old age. There's also the idea that unemployment insurance meant protecting children from the kinds of poverty that could be disabling. And so the prevalence of disability and disability discrimination in the workforce really shaped how people experienced the Great Depression. So a survey done at the time found that 1 out of every 9 people who received public assistance were receiving public assistance because the breadwinner of the family was disabled. And so a lot of what the New Deal does is to create a social safety network that protects us from some of that insecurity. But not all of those protections remain, and the New Deal didn't really create the elaborate safety net that it hoped to. So I think there's still more to do there, and the New Deal most certainly didn't address the kinds of employment discrimination that people with disabilities face. And so even though we've had disability activism and we've had the Americans with Disabilities Act, there continue to be a lot of barriers to opportunity, to employment. There's a lot of discrimination that remains. So disabled Americans earn significantly less than non-disabled peers. They're more than twice as likely to live in poverty. And so if I were making policy recommendations to better position us for the long haul, I would say that one, we need to do everything that we can to reduce the spread of COVID-19 because we don't really fully understand what those long-term implications are going to be. And that number two, we really need to address issues of access and ableism. Having a group of people excluded from opportunity and prosperity isn't just bad for the people that that's happening to, it's bad for the economy as a whole, it's bad for all of us, it undermines growth. And so with that said, the panel's now going to take up a series of questions that focused on herd immunity. And so the first person in this round will be Kevin Williams.
All right, so I'm going to talk a little bit about what we mean by herd immunity and then kind of focus really on the development of vaccines because that's what we're really trying to strive for here. The idea of herd immunity is that you get enough of a population immune to infection or propagation of the virus that basically you can contain the outbreaks. And by doing so, you're able to protect vulnerable people who might not be able to get an immunity naturally or through a vaccination, who would be especially susceptible. We've been able to do this with a number of diseases, things like smallpox. There was enough vaccination that we were able to prevent outbreaks of smallpox because so many of the population is immune to it. Usually the way you get immunity is one of two common ways. One is to get an infection and then recover from that infection and build up an immunity that way. The other is to get some sort of vaccination. And part of what we're going to do in this segment, there's going to be, I think, other people elaborating on why the infection route is not advisable in this particular case. But obviously, there's going to be that risk of further propagating the virus and also the health condition of the individual who's infected. But I'm going to focus more on the idea of getting the vaccine. One of the things that the vaccine does better than our standard infection is the type of immunity the type of immune response that it causes. Generally, there's sort of two parts of your immune response. There's what's called the initial response is sometimes referred to as the innate response. And this is a very non-specific, just generic attempt to get rid of an infection. And so this is things like white blood cells being produced and going out and just trying to destroy whatever the invader is. And it's not, like I said, it's not specific. It also doesn't develop what we call memory. In the future, if you got infected by the same thing, that innate response would be the same, but it'd also be the same if you got infected by anything else. It's not targeted for any particular disease. It's just a broad spectrum, try to have some sort of defense. And that's usually the first round of your response. It usually happens within hours. The longer term response, the one that takes usually a couple of days to get started, is the adaptive immunity. And this is the one that's specific and targeted for the particular infection. And it is something that is going to have that memory. It's going to remember what that infection was like, and it's going to remember how to respond to it. So that's the part of the response you really want to have so that if you're infected again, you can actually respond in the appropriate way. The thing that the vaccine does is because it gives you exposure to something that is not going to cause an active infection, you don't really have to activate that innate response as much, and your body can basically focus its attention on building up that adaptive response. And also because you're not getting sick, you're not propagating it to other people, there's not the health risk associated with it. So really, we're trying to work to get a vaccine so that we can help tailor the body to give a specific response in the case of getting an infection in the future. Now, the question is, how do you know if you've got a vaccine that's effective and safe? And we have a clinical trial process that is active right now, looking at a lot of vaccine candidates. You want to make sure that before you give your vaccine that it's going to work and it's going to be safe. So there's three common phases of the clinical trials. Start with phase 1, and that's usually a pretty small group. You've got 100 or so people in your trial. They're usually pretty healthy individuals, because you're focusing then on, is this vaccine actually safe to give to people? And so the healthy people are less likely to have a really adverse response to it. You're also trying to see, does it cause some sort of immune response? If it doesn't, there's no reason to go on with further trials. So if it gets through that phase 1, it goes on to phase 2 and eventually phase 3, hopefully. In phase 3, you're looking at tens of thousands of people potentially in your study, and you're looking at a diversity in that case. You're getting a lot of ages, you're getting both genders, you're getting races, everything, you're getting diversity across. And what you're trying to see, you're still looking at the whether there is safety, of course, but you're looking more at not only are you getting some sort of response from the immune system, but are you actually being able to prevent the infection? And that's where we are with some of our clinical trials. If you look at some of the stories lately, there have been a couple of clinical trials that were paused in some of the late stages. And that's not anything necessarily out of the ordinary or alarming. What that means is somebody in the trial got sick. They had to pause to find out what it was. Was it caused by the vaccine? Was it caused by some other aspect of the trial, or was it just a fluke that somebody had to get sick from something else? And so a couple of the trials have been paused, and I think in many cases they've been restarted again. And so what we're hoping in the next few weeks to a couple of months, that we're going to start getting some data. And what we need to know is, are these vaccines protecting people against infection? And then how long does it last? Are we going to have to give multiple doses? Are we going to have to give regular doses? That's some of the data that we're waiting for, but we're being optimistic that we're going to start to get some of that data. And if we can get a vaccine that gives us long-term immunity to this, then we're going to be able to get back to normal much faster, because we're going to have that ability to have that herd immunity start to build up, where the outbreaks are going to be far easier to contain if they do occur at all. And so that's kind of the general introduction to this section. And I think Dr. Kessler is going to be taking over next.
And I think correct me if I'm wrong, I have now shared my screen. I hope that is correct. I was so excited to be considered an expert by somebody that I just lost my mind there.
Well, you are an expert, and your screen is shared.
Okay, so these are the mathematical formulas kind of behind the curves that I'm showing you, and I'm going back to those curves, but I want to talk about this just a moment. And give this disclaimer: the percentages that I'm going to show you have nothing to do with our current situation, other than just the way the model responds. So I haven't tried to match real data in these models. I'm just simply showing you how the models respond when we change things. So I want to not worry about all the curves. I just want to worry about the rate of change of infected. So this throwback to anybody who ever survived calculus, you know that when a rate of change goes to 0, that's going to happen at the top of your curve. And so what we want to see is that curve flatten, that derivative, that rate of change go to 0. But then we also want to see it go down, and that means that the slope, is less than 0. So for that to happen, what really needs to happen, there's a little bit of algebra that takes place here. We see this, we need this ratio to be less than 1. This ratio is called the effective reproduction rate, and you may have seen people mention RT in their in discussions. So again, the thing that we really kind of control here is this beta, this transmission rate. And so if we get this transmission rate down, then we get the reproductive rate below 1, and our curve goes down. The other way to make this number small is to make S small. S is the number of susceptible people. And so to make this small with a fixed number in the population, I have to make I big, infected big, or I have to make R big. One of those is a disaster. And so let me get out of this and kind of show this to you. Again, here's the model, and I've set beta kind of low here, so we have a reasonable curve. If I move the number of infected up, okay, you'll see that at some point, the curve never rises. It just starts level and goes down. So right in here somewhere, it looks like it starts level and goes down. Okay, this is a description of one type of herd immunity that occurs in the model. The problem, and this is what Dr. Williams addressed, is this is bad. There's going to be a percentage of these people who do not, they are removed because they die. And this is the situation we want to avoid. So let me do it the other way by increasing the number of removed. Let's say they're removed by the taking of a vaccine. Oh, and let me actually, I want to start this up let me get it to about 20% or so, just so you can see the effect. So if we increase the number of people removed, let's say through a vaccine without getting sick, so you see two things happen. You see the curve is flattening, and that curve gets flatter until it has the same effect, where it starts out kind of peaked and then goes down. So this happened when about 60% or so in this example were removed initially by whatever cause. So so that kept the number of people, it never got above 20%. In fact, it went down shortly after that. That's what we're aiming for with the vaccine, is to get people into the removed category without them ever getting sick. So they're starting in S, they're skipping I, and they're going straight to R. And that's the one that is the best in terms of the care of the people, healthcare. It never overburdens healthcare and things like that. As far as actual percentages for this disease, I'm going to defer to Dr. Eagle about that because I haven't attempted to set these things at any kind of realistic thing for our data. So let me turn it back over to Anthony.
All right. I only have a few things to contribute to this part. I am going to share my screen briefly for a couple of figures. So one of the points that I want to make here is that we need to have a good appreciation for the complexity of the immune response itself. And I'm not an immunologist, so I'm thankful for those people who are and looking at this. What I'm going to kind of give the perspective from the virus side. And so, of course, we've evolved in the presence of viruses, and we have developed a fantastic immune system that helps protect us against infectious disease. But pathogenic organisms at the same time have evolved ways to deal with that. And as I mentioned earlier, the genome of coronaviruses are the largest of the RNA viruses. And it is thought that that's partly part of that reason could be that they have certain abilities to actually deal with our immune system. And a very recent paper that I'm showing here the title of is Imbalanced Host Response of the SARS or the COVID-19 Drives the Development of COVID-19 Disease. So one of the aspects of infection that Dr. Williams just talked about was that initial innate response. And what clinicians have seen and what's been published, there seems to be an inappropriate inflammatory response. And that one of the things that COVID can do is reduce the innate antiviral defense, which part of that innate defense our cells have is to produce a substance called interferon, okay, which is secreted by cells that have been infected with viruses. And this is a way, kind of an alarm system, if you will, to tell nearby cells that have been infected, prepare for this. And this helps the cells get ready and help to defend themselves. But we can see from this publication that the viruses has figured that out and has developed systems to suppress that. In another publication, I'm going to go back a couple here, very recently, and part of the reason I want to show you this is just the detail in which and the speed at which things have progressed, okay? And it may not seem like, to the general public that things are progressing as rapidly because we're still waiting on a vaccine. But these studies are important, and the degree of detail that has been discovered is absolutely amazing. And so this study actually found through cloning of all the genes, okay, these are technology that have been developed and have been applied here, that they cloned all the genes of the virus and looked at the interacting partners from the host cell, and they found that the virus has the ability to disrupt splicing, translation, protein trafficking, among others. So all these processes in the cell are very important for cellular activity, but the virus has developed a way to disrupt those and favor viral replication and making of viral proteins. So in terms of, just simply making the point that is incredibly, we need to look at it both from the host and both from the virus, and we need to look at these things as a unit. One last thing I'll discuss, because of one of the questions that came in, was whether or not there was possibility of reinfection. And there's only a few examples, but I learned just tonight. Dr. Williams said he heard of cases out in Arizona, the possibility of a number of people being reinfected. What I have here is what the original samples or original cases of reinfection. So it looks like there's a small, very small data here, folks. And we may see more of this. And this is an interesting aspect that will need to be studied further. In only 2 of these cases, and there was one in the US, in Nevada, and the other one was in Ecuador, where the patient got a little bit worse or had worse symptoms from the reinfection. And at least with the patient in Nevada, it was confirmed that it was a different version of the virus. So it had accumulated some mutations that made it different enough so they know that it was had mutated over time and or had been a newly acquired virus. We still don't know why, and we're going to need additional examples of this and for us to figure this out. But just to answer one of the audience's questions, or the person who asked this, so it is possible. We've seen a few cases, and there obviously or apparently a number of other cases that have just come out. So this will have to be studied more closely. So I'm going to end there. Thank you very much.
And I'm going to talk about herd immunity from a slightly different perspective, and talk about it from a kind of social perspective, a cultural perspective, and why do we want herd immunity is sort of what I'm getting at here. And one thing to keep in mind is that diseases don't strike people randomly, and I'm referring here to infectious diseases like COVID-19. Disease transmission follows social networks, and I'm simplifying greatly here, but that's really all we need to know to proceed here. So we need to think of ourselves not as individuals, but as people who are connected to other people in social networks and communities, etc. Some folks in our networks, in our communities, are living with weakened or compromised immune systems. Getting COVID-19 is therefore going to be more problematic for them than it might be for others. And once we get to the point where we have vaccines that are available widely, those same folks or different folks might not be able to take the vaccine or receive the vaccine. And we know this because it is the case for many other infectious diseases. Some people simply cannot get vaccinated. So when we talk about herd immunity, part of what we're talking about is being able to protect those folks who are more vulnerable because of either an inability to get a vaccine or because of other issues that compromise their immune system. So what we want to do is build up herd immunity through vaccination when it's possible, because this allows us to do 2 things at once. One, those who do get the vaccine are protecting themselves. And 2, those who can't get the vaccine are protected by being enmeshed or plugged into networks of people who've received their vaccine. So if you get the vaccine once it's ready, it's a 2-for-1 deal. You're protecting yourself, but you're also protecting those in your network who aren't able to get the vaccine to protect themselves. And the other thing that I've been kind of musing on as having a background in anthropology is thinking about the connotations of the term of herd immunity. And what it kind of comes from, or the connotations it brings up, is livestock and kind of mentality of survival of the fittest. And if we think about what we want and what we hope for our families, our communities, our friends, our co-workers, we don't want to have the disease ravage our communities and take the strong toll that it will. We want everyone in our networks, our families, our friends, our relatives, loved ones, community members, co-workers, students, teachers. We want everyone to live and to thrive, right? So we don't want to we don't really even want to entertain the notion of trying to achieve herd immunity through infection because the cost is simply going to be too high. And until we have a vaccine, we really need to protect ourselves and therefore also protect others in our social networks, in our communities, by wearing our masks, keeping our social distance, doing our hand washing, and doing all of those things that we know work in the absence of a viable vaccine.
So we heard from Dr. Williams that we have kind of two paths forward in terms of achieving immunity. You might achieve immunity through infection or through a vaccine. And there have been folks who have argued achieving immunity through infection. But I think Dr. Kessler has shown and Dr. Eagle has shown a lot of people will die if that happens. And so as a historian, I think it's really hard to make sense of death on that scale. And so I think a lot of folks entertain that idea without having a usable past. So I really do think that Americans lack a usable past when it comes to the trauma of disease death on the scale of what we would be talking about. And so if we do have a usable past, it's just many times a number. So in the case of Spanish flu, 50 million worldwide, one of the more conservative estimates, around 675,000 in the US. Again, that's probably a conservative estimate. And so if we even have a number at all, that number is really divorced from the lives that we're talking about. So the lives extinguished. It's, I think, void of the pain of the dying, the grief of the living who are left behind. It doesn't tell us about the consequences of children growing up without fathers or mothers. It also doesn't really give us a sense of the trauma of death on that scale. I think death on that scale is really hard to process. And so I want to spend a little bit of time talking about what it's like to live through a pandemic when public health measures aren't effective or are ignored. And so the war really contributed to the spread of Spanish influenza. And so we see huge spike of infections, and the flu spread really rapidly because of very mobile population and a number of other factors. And so what was it like to live through a pandemic that isn't controlled in the way that the pandemic we are living through? We have a lot of measures in place to control the spread. So there were not enough doctors or nurses. There were not enough hospital beds. They created all sorts of emergency camps, and even there, nurses were overwhelmed and couldn't offer patients the kind of care that they would like to offer those patients. You would see whole families who were sick, and so you had the sick taking care of the sick, children, sometimes even very small children, taking care of parents and siblings. You had the sick caring for so the not so sick, in terms of, not completely delirious with fever, taking care of someone who was completely delirious with fever. So cities ran out of morgue space. There were not enough caskets. There were not enough undertakers. Too many were dying and too quickly. Families were too ill to take care of bodies. There were all sorts of disruptions to services. So in Philadelphia, the city's morgue had hundreds of bodies over its capacity. And so bodies were stacked 3 and 4 high in the hallways in all of the rooms. They didn't have enough ice or ways to control the odor, so they opened the doors to the public. So in full display, these hundreds of bodies over capacity, and one of the things with this particular flu is that many people experienced a lot of coughing up blood and kind of bloody mucus. And so when rigor mortis set in, often there was a lot of blood that would be expelled. So we would be talking about stacks of bodies covered in sheets, bloody sheets. So in New York, the city attempted to stop the spread by banning spitting and promiscuous coughing and sneezing in public. Communities tried to use public spaces like schools for hospitals. Some set up tents and field emergency care spaces. Some hospitals didn't turn patients away even if there were no beds, so you saw patients in the floor. Cities across the country staggered factory shifts and store hours. If theaters weren't shut down, they reduced capacity significantly. So in Bellevue in New York, patients flooded the hospital. So a doctor there at the time was responsible for about 500 patients at any given time. And she said that the patients were given aspirin and whiskey, and that was really all that could be done for them. And she was so overwhelmed with patients that she typically only saw them twice. So when they were admitted and when she signed their death certificates. Bellevue had so many patients that patients shared beds and slept in the floor. Others had to rely on family members or community members for care. So nurses really played a central role in caring for the sick, and nurses really saw some of the most terrifying moments of people's lives. And so just for example, a Philadelphia nurse found a woman who had given birth to twins, and her husband's corpse was in the room, and she had been alone for about 24 hours without being able to, no one could hear her call for help. And in Chicago, a nurse found a little boy in the street who was in his nightshirt and completely delirious with fever. And so she helped locate his home, only to find that every single person in the home had influenza with fevers of around 104 degrees. Another nurse in rural North Dakota talked about encountering a family, and the mother was so delirious with fever that she had barricaded herself in a room and was perspiring like the rain. And her children, she had all dressed them all in their finest clothes and kind of laid them out. I think perhaps thinking that someone would find them that way, no longer alive. And so this nurse arrives to find children with fevers ranging from 102 to 105. So everywhere, communities were overwhelmed with illness. It wasn't just something that happened in cities. It wasn't just something that happened in a few places. Basic services, infrastructure was really challenged. So flu spread through police officers. So it was hard for cities to have enough police. It spread through the ranks of firefighters, of garbage collectors, of grave diggers. The telephone company, Bell Telephone Company in Pennsylvania, had so many cases that they had to restrict phone calls only to emergencies. And so I could give you examples of this kind of trauma and terror. But we're told over and over again about the dangers of overloading our healthcare system, and that's true and that's important. Those strains can also disrupt care for other people, maybe people who aren't critically ill, but could become so. But uncontrolled pandemics also disrupt functions like caring for the dead, police, fire, garbage collection. They create strains on families and communities that sometimes those units can't withstand. And so we've focused a lot on the economic cost of public health in our discourse. But death has costs as well. And, I think it's important to remember that infectious disease, it can strike the rich and poor alike, but people who are already struggling to make ends meet have historically suffered more during pandemics and epidemics. So illness can be compounded with food insecurity, housing insecurity. And so people who are marginalized and already struggling to access care have experienced disproportionate amount of illness and death. And so those are important factors to keep in mind. And so with that, I'm going to turn it over to my colleague, Rodney King, who's going to talk a little bit about testing.
Okay. Very quickly. All right. So most of you are probably aware of a couple of different testing techniques that are being used. I looked at the emergency use authorization on the FDA website today. There are over 187 tests, mostly which are based on this technique that I'm showing you right here, called the polymerase chain reaction. So what this technique allows you to do is amplify pieces of nucleic acid, okay, and what I'm showing you here is DNA, starting with a very low amount and then amplifying, making much more, many more copies of that. And the basic principle is that DNA is double-stranded, and we can denature or pull apart those strands, okay? The DNA is complementary to each other, so they kind of zip up, and then we can denature, and we do that simply by heating. And so the two strands of DNA will come apart. And then we'll cool that reaction a little bit. And in the presence of these things called primers, as indicated here by the orange boxes, these are simply pieces of nucleic acid that you can order, you can synthesize, and they will be complementary to a sequence that you're interested in. And then we'll find its partner and zip right up with that. And then together, the next step would be the extension of this particular primer and copying of the template DNA. So there are enzymes present and nucleotides, okay, the bases of DNA that are present. And you would extend that and copy the other strand. So in the end, what you have is going from one molecule over here on the left to two molecules over here on the right. So that's the whole idea, that you've amplified this, and you do this again and again and again, and you get millions of copies of this in a matter of an hour or whatever. So it just depends upon the technology that you're using. Very powerful, allows you to use very small amounts of starting material that you wouldn't be able to detect otherwise, and then be able to detect it by this particular technique. Now, the technique that's actually used for this particular virus is called Rtpcr, okay, reverse transcriptase Rtpcr. And so what I haven't shown in this diagram, there would be a step over here. There would be the RNA of the virus. And that RNA would have to be first copied into DNA. And so this technology was developed after the discovery of an enzyme called reverse transcriptase. And reverse transcriptase allows you to copy RNA into DNA. And then once that's done, after that step is done, you can just simply do the steps as I've shown here. So this is the basic protocol, or what's used to, in fact, detect whether or not you have an active infection, that is, or you have you have an infection of COVID, through taking a sample, a nasopharyngeal sample, and it doesn't take much virus in that case, and then you're going to amplify that many times to see if you have it present. So that's the most common one. Another type of testing is based upon antibodies. And so in this case, we have, and this is a rapid test. And in this case, we have antibodies. So the virus has been, or viral proteins have been purified. Antibodies have been made in animals, and those antibodies have been purified and then labeled some way that we can see the reaction afterwards. But here, imagine the analyte shown in this figure is the virus, okay? This is deposited on basically it's a paper that will, once the sample is deposited, it will wick along and move along, very similar to what happens with a pregnancy test, for example. And so that moves along. If the antigen binds to the antibody, that whole complex will move along. And then farther down the strip, there are antibodies that will bind to that antigen as well. And that will capture this complex. And once that's captured, that's how you get the development of that line that you've probably seen or heard about. So the secondary, or that is, there's another line that just simply confirms that the test is working. So anything that was not bound goes to some of the antibody in the second line. And this is important because it shows you that everything has moved along this strip and that you get a positive, or you get a line forming there telling you that the test worked. And therefore you can be confident whether or not your test line actually shows up or not. Okay. So this is the rapid test. It's based upon antibodies that are going to be specific for the virus. Okay. And we're just exploiting that property of the protein in order to specifically detect the virus. Okay. So it's not as sensitive as the Rtpcr, because the Rtpcr, of course, again, amplifies the signal many, many times. Now you do get some increase in signal here because you can have, sometimes you can have an enzyme there or something to catalyze a reaction to give you a colored product or some other way of visualizing this. And so that does increase the signal, but it's going to be nowhere near the Rtpcr. And then finally, you can actually detect whether or not a person has had a previous exposure to the virus by detecting antibodies that they've made. And again, that's based upon antibody technology. Same kind of concepts as shown here. So that would only tell you whether the person's been exposed and they've produced antibodies. So the rapid test you hear about is what I've just shown here, and the Rtpcr is what I showed in the previous slide. So those are the basics of how and what's going on with the testing and how that is done.
Okay, so I'm going to talk a little bit about what you should take away from getting a test and what you should do once you get test results. So say you've been tested with one of the two diagnostic tests, either PCR or the rapid test, and it's negative. So what does that actually mean? What it means is that at the time you were tested, which may have been a couple days ago, maybe just a few hours ago, you didn't have an active infection. It doesn't mean that you can go ahead and stop social distancing or mask wearing or being cautious. You still need to maintain those behaviors, because remember that this is a disease that is transmitted one person to another. So having a negative test does not mean that you have any sort of immunity to the virus. It just means that at the time you were tested, you did not have an active infection. Okay, so let's go back to that same scenario. Say you've been tested, but it's positive. What should you do in this case? What does that mean? It means that you do have an active infection, and it means that you need to isolate. Your case will be reported to the health department, to the public health department, and you'll be contacted by a contact tracer. A contact tracer will talk to you, will get some information from you, will share resources with you, and importantly, will ask who you've had contact with. And this is really important because, like I said earlier, diseases follow social paths, right? And I am simplifying, but the contact tracers are asking who you've been with, not to determine if you've done something you shouldn't have done, if you've been at a gathering with more than 10 people, for example. What we're trying to do with contact tracing is, again, get ahead of the disease, and we're trying to head off the spread of the disease or any outbreaks. So what they'll do is contact those that you've been in contact with, check in with them, ask them about symptoms, give them resources. I talked about whether or not they need to go into quarantine or isolation, whether they should be tested, etc. So if a contact tracer calls you, you're not going to get scolded. Your medical information is not going to be shared. Your privacy, your confidentiality are going to be respected. All this is one of our tools to get ahead of the disease because it is spreading fairly rapidly. Another thing to keep in mind with testing is that when you've been tested, you should isolate while you're waiting for your results. So if you test positive, that positiveness kind of goes back in time. And so you haven't been negative until you get the results. You've been positive from the time you had the test. So if you have to wait a day or 2 days between when you take your test and get your results, you do need to remain isolated, because if you are positive, you could potentially be spreading the disease. And contact tracers are your friends. They are wealth of knowledge and can give you information that you need about how to deal with the disease and how to quarantine and isolate successfully.
All right, so I'm going to talk a little bit about some of the challenges of drug design and some of the things that people are looking into right now to try to come up with some sort of therapeutic agent that can help people who have been infected. So the challenge is, anytime you're trying to design a drug, you're trying to stop some sort of invading organism or stop some sort of rogue process without causing collateral damage. You don't want to harm the processes that are happening that are good. And so that always gives you a little bit of a challenge. You have to be fairly specific. And the other thing with viruses, they're particularly hard to treat because they just don't give you much to target. They're pretty simple. They've got some genome in there, and then they've got these viral or these protein codes, and there's just not that much to target. But there are 2 or 3 things that people are looking at right now as possible targets for drugs to help treat against COVID-19. One is to actually look at targeting those viral proteins. So if you've seen the picture of the coronavirus, or if you remember when Dr. King was talking about those spike proteins, that's something somewhat unique to these types of viruses. And so there are people looking at ways to interact with those spike proteins and potentially block their interactions with the cells. Generally, their binding, as he was talking about, to these receptors and co-receptors. And so if you can block that interaction, you could potentially prevent the virus from being taken up into the cell and then propagating from there. So there are some people looking into those sorts of treatments. Another possibility is to look at blocking some of the downstream processes that cause the disease state. There's been some talk about what is called the cytokine storm, and more recently people have been talking about something called a bradykinin storm. This is a small peptide that causes signaling that, when it gets out of control, can lead to leakage of blood vessels and buildup of fluid in the lungs, and some other things that are at least consistent with COVID-19 symptoms. And so people are now looking at, can you block the bradykinin receptors to prevent this excess of bradykinin from producing some downstream effects, if that is part of the issue. And then a third thing that people are doing is to try to look at ways to block the genome from being replicated. If you remember the cycle that Dr. King was showing, the virus gets inside the cell and uses the cellular machinery to try to replicate its own genome. And so one of the tools that's been used there is this drug you may have heard of called Remdesivir. This is something that recently got FDA approval. It's not a cure-all, but it is helping against the propagation of the virus in patients who are infected. And what it does is it helps to try to block and slow down the replication of that genome so that the progeny of the virus are potentially not as effective at spreading further disease states. So those are 2 or 3 different strategies that people are looking at ways to come up with something that will blunt the effects of the virus so that people either don't get very sick, or if they do get sick, that they can recover. And so hopefully, as the next few months go on, we're going to strategies, new drugs are going to be approved, and together they can start to really help people who are very sick to get better, so that this doesn't have the long-term effects and the mortality that we're currently experiencing.
Hi, so I'm going to very quickly address a question about the economic impact of public health efforts. And so I want to respond to this question by kind of turning it on its head in 2 different ways. So one, I think a lot of our discussion about public health efforts and the economic impact of public health is based on the assumption that public health in some way harms the economy. And so I want to kind of point out some examples from history that public health actually can create jobs and can be really good for the economy. So during the New Deal, there was a huge investment through programs like the WPA or the PWA in building public health infrastructure. And so in Kentucky, that was really, really important. So what we see in Kentucky is there's this massive investment in building water supply systems, water treatment plants, sewage treatment, and installing 100 miles of water mains, so improving the water systems. There were also over 50,000 sanitary toilets built in rural communities where kind of city sewage didn't make sense. And basically, the state commissioner of health credited this work with the almost entire elimination of typhoid fever and waterborne disease in 120 Kentucky counties. And so I think that's kind of one piece. The New Deal spent a lot of money expanding hospital spaces. Kentucky saw a 20% increase in hospital capacity. Kentucky's TB sanitarium saw a doubling of capacity. The New Deal also worked on sealing mines that were leaking acid and killing vegetation and destroying fields, and destroying streams and waterways in Kentucky. And so there was a lot of effort on education, visiting nurses, providing vaccinations, providing health care to children, people living in poverty, efforts to eradicate disease-causing pests. And so essentially our health commissioner said, like, these projects were of incalculable economic value to the state. So we're talking about saving lives, but also creating jobs and building infrastructure for Kentucky. And so I will bring up that example because I think that we need to stop thinking about public health as only causing difficulties. And then the second piece here is that if we focus only on the costs of public health measures to the economy, I think the thing that we're ignoring in that piece is that death has real economic costs. And so uncontrolled illness also has real economic costs. So the Spanish influenza pandemic destroyed families, exhausted savings, and created incredible hardships, not because of restrictions, but because of uncontrolled illness and staggering amounts of death. And so families then, as today, rarely had enough savings to weather prolonged illness, or to cope with a family member who was a primary wage earner passing away, or to deal with the cost of burying multiple family members. And so many families struggle to bury their dead. And so I want to tell you just two stories and then I'm going to turn it over. So both of them are about teenage boys in New York. So one Bellevue patient, just a teenager, saw all 10 of his family members pass away from the pandemic. And he faced a choice of saving the family's business or burying his family members. And he was in the hospital because he had also been ill, and talked to his doctor, and his doctor recommended letting his family members be buried in Potter's Field and to save the business. So the New York Times made an appeal on behalf of another boy who was just 14. And so they were fundraising on his behalf. And so his father had died from influenza, and he was seeking, working in a factory, trying to provide for his family of 8 and struggling to make ends meet. One of the children went into foster care, and he still couldn't manage to feed his siblings. That father too was buried in Potter's Field. And so again, I think we need to think about the cost of public health differently. Public health can create jobs and build infrastructure, but I also think that death and illness have a lot of costs that we are seeming to ignore. And so I'm going to turn it over to Bruce.
Hey, Bruce, your mic's muted.
Every iteration I mess up. I apologize for this. So very quickly, I just want to make the point that when we watch new cases, we've seen new cases rise, we've seen new cases fall, we've seen them rise again. And this doesn't really kind of fit the model that I've shown you. So I want to make the comment that the models have made the assumption that our transmission rate is a constant over time, and it's actually not. And so to kind of demonstrate this, I just want to look at Kentucky data. This was a problem I gave on the second test in my math modeling class this semester, where I gave them new case data. And so this is actual new case data for Kentucky. And the blue line shows that how the data kind of sometimes fluctuates over weekends and things like that and catches up. So the darker line shows kind of a smooth version of the light blue. And this is actually not the curve we're trying to flatten. The curve we're trying to flatten is the number of people infected at a given time, but it goes along with this, right? You can deduct what the transmission rate is from the number of new cases, and you can generate the infected curve from this. So what do you take from this? Well, we don't want to be here right now. That's clear. And this is, in a way, reflected. You see similar shapes in the new cases and the number of infected. This is a calculated number of infected people, by the way. When we see these things go up, they're reflected in both graphs. I can tell you what some of these days correspond to. The 100th day from March 26th, which is when the governor started releasing the daily data, is July 4th. So July 4th was here, and we see things shooting up. The 200th day or so is about 10 days after everybody got back from fall break. Not necessarily I'm not giving causality, I'm showing you correlation. So, but I am trying to show you that these models, even though they are abstract in some sense, can be applied to real data and can be correlated with events that occur on the calendar. So I will turn it back over to Anthony.
Thank you, Bruce, and thank you to each one of our experts. You know, I think we have just a few minutes here before our planned end time. And so, you know, I think, you know, one topic that's often a common point of discussion is, you know, kind of let's talk about the impact of mask wearing, both from a public health standpoint and how that then kind of affects everything that we're kind of seeing in Audra from a historical perspective, the role that mask played in the Spanish flu. You want to get us started, Audra, from a historical perspective?
Sure. So I think that in terms of masks during the Spanish flu, the Red Cross was really encouraging masks. And so a lot of the nurses who were traveling and visiting folks were wearing masks. We see a lot of images, I think a lot of the images that we see from 1918, 1919 show masks. I think folks started wearing those after we have really widespread community spread of influenza. And so, you know, I think that folks had to be convinced to wear masks. We didn't have necessarily the same mask mandates necessarily. But there were a lot of mask wearing taking place in like army camps, training camps, a lot of nurses wearing masks, and that sort of spread over. But when we look at kind of what are those key public health recommendations at the time, masks wasn't really one of them. So folks were encouraged to avoid students coughing and stop spitting in public. Folks were encouraged to wash their hands and make sure to be well rested. So the idea was, if you're worn down, you might be more likely to get ill because there's not as clear a sense of how one might protect themselves.
Thank you. So, Dr. Eagle, you kind of spoke to kind of, responsible public health measures, and, and I think this, many would agree that, it's probably one of the greater challenges that we've had is kind of convincing folks that those safety measures have positive outcomes. So you want to speak to the role of public health and helping educate folks about the value of wearing masks?
Yeah, so this is something that we all sort of discussed as a group, but I don't think it made it into this panel discussion. How what we're seeing through this pandemic is science unfolding in real time, which is not, and the whole process of the sort of self-correction that happens in science is happening in the public eye, which is not something that usually kind of non-science folks pay attention to, because by the time an article has been published and been through review and all of those processes, we're pretty certain of things, right? However, we're all learning in real time, and so a lot of the hesitation about mask wearing maybe stems from the sort of back and forth early in the pandemic about whether or not masks were valuable, whether or not they were useful. The thing is, now we know that they are. The science is sort of caught up with where we are, and we know that masks do two things. They protect you, the wearer, from contracting COVID-19, and they also protect you from sharing COVID-19. So it's the responsible thing to do for two reasons. One, it protects you. Two, it protects your community. So for that reason, it's a win-win situation. So we do want people wearing masks for that reason, not only to take care of yourself, but also to protect others. And we, while everyone witnessed kind of the sort of halting steps of understanding how masks are functioning in this pandemic, we all saw that happen in real time, but we now do understand broadly that masks work.
Thank you, Dr. Eagle. Dr. Williams, King, Kessler, any parting thoughts that you all would like to share to add to that topic?
I think she summed it up nicely.
So it is, I do agree that it has been interesting. And Audra, I think we'd probably talk for hours about, had social media and a 24/7 news cycle existed during the Spanish flu, how different would it have been? And so I do think that, I hope that each of you and your students have taken advantage of the opportunity to learn from you and your expertise kind of in real time as this is unfolding. And hopefully what is in once in a lifetime scenario that they will incur. Definitely thank you all for taking time to share your knowledge and your expertise with our alumni and friends who have joined us this evening and who watch the recordings that will be available afterwards. Thank you for the work that you do in supporting our students here on campus. I know our faculty have totally innovated learning in this environment, and and our students as well have adapted to that. So thank you each to you for what you do here at WKU, for sharing your knowledge with our alumni and friends, and I wish you a successful end to the rest of this semester. Be well, be safe, and we hope to see you soon. So have a good night.
Thank you.
Thank you.